Fungus. Even the word sounds gross. However, from mushrooms to molds to yeasts, fungi are actually pretty cool organisms. And without them, we wouldn't be able to eat portobello mushroom burgers or drink beer or wine. But they're not so cool when they cause infections, like athlete's foot, yeast infections, thrush, and toenail fungus. In addition to these relatively mild fungal infections, more severe systemic fungal infections are becoming more common in immunocompromised patients, such as those suffering from AIDS. These patients can get opportunistic infections caused by fungi that are around us all the time but are normally harmless. So it's definitely very important to have good antifungal drugs. The problem is that fungi, being eukaryotes, are very similar to our own cells, so it's hard to find drugs that are selectively toxic. Unfortunately, there aren't that many antifungal drugs available today, and the ones we have can cause major side effects, so they're often only used for infections that are really life-threatening. In this lesson, we'll look at the major types of modern antifungal drugs and see how they attempt to selectively target fungi without harming our own cells too much. Remember that one of the major targets for magic bullets, or selectively toxic drugs, was the microbial plasma membrane. But wait a minute, all cells have plasma membranes, and eukaryotic cell membranes must be pretty similar to each other, right? So how could a drug only target fungal plasma membranes and not our own? The answer is that fungi and animal cell membranes have different sterols, which are special lipids that can regulate membrane fluidity. You've probably heard of cholesterol, which is the major sterol in animal cell membranes. Fungal membranes have ergosterol instead. So many antifungal drugs target ergosterol as a way to disrupt the fungal plasma membrane. These drugs make fungal membranes very permeable, meaning that cellular contents can leak out and molecules from the outside can get in. This causes the fungus to die, so these agents are usually fungicidal. Polyenes, such as amphotericin B, are important drugs for treating systemic fungal infections. They attack ergosterol, injuring fungal membranes. Amphotericin B is an ingredient in topical preparations to treat thrush, which is a yeast infection in the mouth. Remember that when a drug is used topically, it means it is only used on a small region of the body. In contrast, giving a drug systemically means that the drug is distributed throughout the whole body. Amphotericin B is pretty harmless when used just in the mouth, but when given systemically as an IV, this drug is very toxic, especially to the kidneys and liver. It's so toxic that it is sometimes nicknamed amphoterrible. This drug is given as an IV only in extreme cases, for example, systemic fungal infections. The next group of antifungal drugs is the azoles. These drugs inhibit the synthesis of ergosterol, which means they block the synthesis of the fungal plasma membrane. A couple of these drugs, like clotrimazole and miconazole, are mainly used topically, for example in creams to treat athlete's foot and yeast infections. Another related drug, ketoconazole, can be used systemically and is often used as an alternative to the more toxic amphotericin B. Finally, fluconazole is even less toxic and even more water-soluble. That means it is easier and more effective for systemic fungal infections. Another option for selectively killing fungi is to target their cell walls. Remember that animal cells don't even have cell walls, and fungal cell walls contain certain unique molecules that are not found in any other organisms. The echinocandins are a relatively new class of antifungal drugs that prevent fungi from making beta-glucan, an important component of their cell walls. When a fungus is treated with an echinocandin, it makes an incomplete, weak cell wall, which causes the fungus to lyse, or burst. This is similar to how penicillin kills bacteria. Flucytosine is another commonly used antifungal drug. It is fungistatic because it blocks DNA and RNA synthesis and thus prevents the fungus from making the new DNA and proteins that it needs to reproduce. How does this drug not affect our own nucleic acid synthesis? It's actually pretty cool. Flucytosine is a prodrug, meaning it's not toxic at first. Inside fungal cells, an enzyme converts flucytosine into 5-fluorouracil, which is the active drug. Our cells don't have this enzyme, so we aren't affected. Unfortunately, fungi can rapidly develop resistance against flucytosine, so it is usually given in combination with another drug, like amphotericin B, to make drug resistance more difficult to achieve. One last antifungal drug is griseofulvin. This drug inhibits microtubules, which are required for mitosis. Therefore, griseofulvin prevents fungi from reproducing. Apparently, animal cells don't take up griseofulvin as efficiently as fungal cells do, and this may be the reason for its selective toxicity. Griseofulvin is mainly used for fungal skin infections like toenail fungus and athlete's foot, because even when given systemically, it reaches high concentrations in areas of the body that contain keratin, like our skin and nails. Let's review. We've learned about 5 different types of antifungal drugs that are used today. First, we learned about 2 classes of drugs that target fungal sterols, which are special lipids that can regulate membrane fluidity. Our cell membranes contain cholesterol, but fungal cell membranes contain ergosterol. The polyenes attack ergosterol and injure fungal plasma membranes, and the azoles inhibit ergosterol synthesis, blocking fungal membrane synthesis. Toxicity of these drugs is a major concern, especially with amphotericin B, which is so toxic that it can only be used systemically in extreme cases, such as life-threatening systemic infections. Next, we learned that the echinocandins prevent fungi from making beta-glucan, an important component of their cell walls. These drugs work the way penicillin works against bacteria, by weakening the cell wall so that the fungus bursts and dies. We next looked at flucytosine, which blocks DNA and RNA synthesis and is specific to fungi because the enzyme that converts it into an active drug is only found in fungal cells, not our own cells. Finally, we learned that griseofulvin inhibits microtubules, which are required for mitosis. This prevents fungi from reproducing. Griseofulvin is unique in that it reaches high concentrations in areas of our body that contain a lot of keratin, like our skin and nails. Thus, it is often used for infections like athlete's foot and toenail fungus.